Professor Chris Yu from Cornell University. He comes to us from it's actually the School of Applied Engineering Physics. Here we go. And as I mentioned, we're probably going to move up a little bit different scales from molecular to more the cellular. And Chris is an expert really on combining a lot of the engineering in fiber optics and source technologies with this bioelectrical imaging and biophotonics. He's also, I guess, the new associate editor for Biomedical Optics Express, so he's a good person to get to know for as you're trying to get things published.
We're doing it for a year, right?
And also a fellow of Optical Society of America.
Okay. Thanks, Stephen, for the introduction, and it's great to be back. I gave a talk last year at the Material Science Research Center. So this probably is more appropriate. Brain imaging with multiphoton microscopy. You can interrupt my talk at any time you want. We can interact, so make sure everybody understands. My group essentially does three things. Brain imaging, which we'll talk about today. We also make miniature multiphoton microscopes, and you will see perhaps someday this will also be useful for the brain. And we do fiber optics, so still having great fun, my legacy of the industry work. My students, postdocs, and collaborators, support from a variety of agencies. So here's a brief outline. I try to motivate why do we need to go deep into the brain. And I focus on the technology development for brain imaging. If you think about the brain initiative, even though it's neuroscience centric, but on the other hand, it's really all about tool making. What kind of tool is necessary to look at brain structure and function? So look at 1700 nanometer imaging, 1300 nanometer imaging, give you some examples. And I'll end the talk by discussing some future directions, tell you where I think we are at and what we think we can do further to advance this field. So here's the motivation. This is the status we are at for the neuroscience. On the one hand, we know pretty well, very quantitative input outputs at the level maybe, say, a dozen neurons. We know this pretty well by doing optical imaging, by doing patch clamp, electrical recording, and things like that. On the other side, we can observe behavior, like a mouse running on a treadmill, seeking water, seeking food. We have some knowledge about that. Quantitatively, you can see, okay, we can see, for example, about 100 neurons imaging about this deep, maybe say 700 microns, a few hundred microns across. That's the field of view. If you look at the mouse brain, it's about a 1 centimeter across. The volume is about 1 cubic centimeter for a mouse brain. This yellow box is roughly what we can do right now. That's all we can see with a resolution of a cellular level or subcellular level at a micron resolution. That's the kind of volume you can detect. So about 6-7 years ago, I looked at my group's direction. I thought, hey, can we do something more than this? Can we enlarge this area by going much deeper and hopefully also much bigger volume? Right? So we can study like computational columns, things like this. To do this, you really have to new tools to bridge this gap of a sort of a microscopic observation, sort of a macroscopic observation. And this tool, of course, inside of optics is really doing optical. Because optical is really great for look at brain activity because optical is non-invasive. Optical also provides high spatial resolution and also high time resolution. So all these are very good to in vivo monitoring of biological dynamic phenomena. So again, you need fast and deep tools imaging the brain, hopefully not damaging it. So the tool I use is called multiphoton microscopy. And I worked on this, just like Jixin, more than 20 years ago as a graduate student in Walt Webb's lab at Cornell. So this is like the diagram shows literally more than 20 years. So this is a molecule, ground state and excited state. So instead of Raman excitation, here we do fluorescence excitation. So here what you do is a molecule absorb, for example, a blue photon, make a real transition to the excited state and a vibronic decay and say green fluorescence. That's your 1 photon absorption and 1 photon fluorescence. But the twist for multiphoton is that this same molecule can actually absorb 2 photons simultaneously, in this case, say 2 red photons, to make the same transition, roughly to the same excited state. And again, vibronic decay and a green fluorescence. So that's what it is. The only trick is that if you think about one photon absorption, the cross-section, or essentially is how probable a molecule gets excited via this one photon transition, is about 10 to the minus 16 centimeters squared per photon. I'm not sure you're familiar with those units, but it doesn't matter. What I want you to see is this. For one photon transition, this is the probability. For two photon transition, you can see the probability goes much lower, because here you have a virtual state, and virtual state has a very short lifetime. That's essentially how Raman works. That's why Raman's cross-section is very tiny, right? So the point is that the two photon cross-section, or the probability of exciting a molecule via the two photon process, is very low. So to make this happen in a reasonable way, to make it practical, you need a femtosecond source. That is, when the laser is on, the laser is very bright, 1 billion times brighter than the surface of the sun. But most of the time, the laser is off. So by doing so, you can actually make this weak process a practical contrast for imaging biological sample. And for example, if you do two photon excitation, the probability is proportional to the inverse of the duty cycle. The duty cycle is the frequency or the repetition rate of your pulses and the pulse width. Okay, that's a duty cycle. So we can make this practical. And a physical example is like a fluorescein molecule. Fluorescein is very ubiquitous, very cheap, and you can also drink that, FDA approved. You just turn green after a few minutes, but that's not too bad. You have a fluorescein molecule, this is wavelength axis. You can do one photon excitation, blue photon coming in absorbs, green photon emission, very simple. And you can do two photon absorption, simultaneously absorbing two red photons. Now red photon, as you can see, is longer wavelength than the fluorescence emission, right, because of energetics. And you have the green fluorescence emission. So in that regard, looks very simple. One photon, two photon, almost the same thing. So what's the big deal? So this is actually one of those slides, one of the slides convinced me to join this field when I first met Walt Webb 23 years ago, right? He showed me basically this slide. See the wonder of one photon excitation versus two photon excitation. If you do one photon excitation, the fluorescence signal is proportional to the intensity. That means in addition to the focal plane where you want to have excitation, you also have excitations above and below the focal plane. Very simple, because you just double cone tracing out the illumination path. That's one photon fluorescence. If you do two photon fluorescence, since two photon is needed to make this transition happen, okay? That means the signal, the fluorescence, is proportional to the intensity squared. When something depends nonlinearly, what does that mean? That means the fluorescence is only appreciable at the focal point, because the focal point has the highest intensity. So if you look at this same fluorescent cuvette, only one point, I don't know if you can see in the back, only the focal point is bright. So you only excite the focal point. There's no fluorescence above or below the focal plane. So what's the big deal, right? The big deal is that if you want to study tissue, if you want to look at cells in the native environment, say neurons in the brain, you got the image through tissue, right? You will see the competing technology at the time, say confocal microscopy, doesn't really work with tissue. So let's go through some details here, right? So let's look at how confocal achieve three-dimensional spatial resolution. So this is one photon excitation, as we talked about, right? One photon excitation we talked about. You have fluorescence from the focal plane, fluorescence above and below the focal plane. Now this focal plane position, this focus, is the only place where you have high spatial resolution information, because the light spot is very tiny. So what confocal does is that it puts a pinhole before the detector. So only the fluorescence from the focus is going through this detector and being detected. That means even though I excite the fluorescence everywhere, but only the focal point fluorescence can reach my detector. So essentially by doing a spatial filtering, you can sort of selectively detect the fluorescence where you want. Okay? If you do two photon, since all the fluorescence is coming from the focal point, as we just talked about, intensity square dependence, that means I don't need this spatial filter. I collect all the fluorescence coming back. So in this regard, looks pretty simple. It's like, okay, no confocal pinhole is needed, but what's the big deal, right? The big deal is that if you want to study tissue environment, every cell sits in some kind of a tissue if you want to look into that native environment. So if you don't have tissue, don't have scattering, you can see fluorescence from the focal point will be faithfully imaged onto the pinhole. Everything works. That's confocal microscopy, right? It all works. But once you have tissue, what tissue does is to scatter the photons. That means even though the photons are coming out of focal point, as they propagate back, epi collected, it gets scattered by the tissue. Every time the photon gets scattered, it can change its propagation direction. When that's happened, you can see you can no longer faithfully image a point to a point. So in this case, at the detector plane, instead of having a single point, like over here, you actually have an extended illumination pattern. It's very big. If you put your pinhole over here, you can see all the fluorescence, most of the fluorescence gets blocked, can no longer be detected. Even worse, even if fluorescence coming above the focal plane can actually have a possibility of getting scattered back into your pinhole, right? So it took about 5 years for people to realize this multiphoton microscopy is really useful. That's about 1995 at the time. Is for imaging deep into scattering tissue. Because for multiphoton microscopy, you don't need a pinhole. Everything is coming from the focal point. I collect all the light, and it all works. Even though the photon gets scattered, if my detector is big enough to capture this scattered photon, the signal is the same. So this deep tissue penetration is really the main advantage of multiphoton microscopy. Again, it took almost 5 years for people to latch onto this. And in addition, as I mentioned before, if you do 2-photon excitation, now we do 3-photon excitation, you use much longer wavelength. And longer wavelengths typically reduces the effect of tissue scattering. That's why the sky is blue. That's why the sunset is red, right? Scattering versus function of wavelength. If you put these two factors together, you will find the collection advantage, because we use a big detector, no pinhole needed, we gain roughly a factor of 100 if you want to go, say, a few scattering depth into your scattering tissue. And if you put this long wavelength advantage also into account, you gain another factor of 100 also, so you get a 10 to the 4th. Yes?
What about scattering before the excitation part?
Yes. So scattering for the excitation light, that you still suffer. You do. But on the other hand, it suffers a little bit less because of the longer wavelength used. So that still happens. But suffering at the excitation path is much better than suffering at the detection path. The detection, the photons, the signal photon is the most precious, right? The illumination, in some sense, you can always increase the laser power somewhat to get a little more illumination. But signal is really associated with excitation, associated with photo damage and all those type of things. Make sense? So now multiphoton is really good, right? 2-photon has made a great advance for neuroscience, for example. But how deep can we go? It cannot go infinitely deep, obviously. We have to look into this issue called signal to background ratio in scattering imaging. This sort of comes back to your question about the excitation scattering. So let's first look at this. So this plot looks at the intensity of the light at the focus as a function of imaging depth. So as you illuminate the sample, the red denotes the illumination. If the tissue is scattering, say this is scattering tissue, that means every time an excitation photon encounters a scattering event, the photon essentially will be removed from the excitation path. Again, excitation changes the propagation direction of the photon. If you look at this process, essentially only the ballistic photon can reach the focal point. And the intensity of the ballistic photon as a function of depth, focal depth, goes down exponentially. Okay? This is essentially the same statement as every time photon gets scattered, it's removed. And that process is like a Beer's law process, is exponential process. Okay? So this is its imaging depth, that's Z, and the LS is the scattering length, meaning scattering length. Okay? So the intensity of the focus goes down exponentially. Okay, that's a scattering effect. In addition, also we know, we focus the light to a focus. And that means the intensity at the focus also gets enhanced by the geometric focusing event, focusing effect. Right? So if you put these two effects together, you have the exponential decay, that's due to scattering. You have the geometric focus, gets you high intensity. So eventually you see an illumination profile looks like this, right? This is the focus, this is the scattering, okay? So you have two functions multiplied together. So you can see as you go deeper and deeper what happens. That means you have more and more illumination at the sample surface and less at the focus. So eventually what happens is that this is the signal what you want, but you also generate a lot of stuff above the focal plane because of this exponential decay. And eventually you go deep enough, this background, which is above your focal plane, can overwhelm the photons from your focal point. And this is what I'm saying, that if you go deep enough into scattering tissue, even if you do 2-photon excitation, it's no longer three-dimensionally localized because of the exponential scattering process. Okay? And you can put this roughly in mathematics. You can look at signal, which is at the focus, divided by all the light above the focus. You can look into these two ratio, that's called a signal to background ratio. Okay? And this thing goes like this for 2-photon excitation. This is the scattering effect. This is the geometric focusing effect, the power law effect. So you can see this is your signal to background ratio. As you go deep and deep into the sample, you can see Z gets larger. What will happen? Right? This is the power law, this is exponential. Of course, exponential function always wins. Exponential function means power to the infinite power, right? That's what exponential function essentially means. And that means you cannot really go that deep because eventually this guy goes to zero. That means everything you collect at the sample surface. So that's the fundamental limit of how deep you can go into the scattering tissue, even if we do 2-photon excitation. So the strategy we've been pursuing the last few years is doing two things. One is to try to go even longer wavelength to reduce the effect of scattering. Essentially is to make this decay length much gentler, okay, much longer decay length. Another is if I can make the confinement or the three-dimensional confinement of the focus even better. That's going to high order nonlinear process. So let's go through a logic here. So if you want to reduce the effect of scattering, going to long wavelength is almost no-brainer. I think pretty much everybody knows. If you look at this is the scattering coefficient. All these curves are mimicking the mouse brain, but that's what we're focusing on, okay? So if you look at the mouse brain scattering coefficient, this is a function of wavelength. Oops, what happened? As you can see, as you go to longer wavelength, the scattering coefficient decreases. That means the scattering length increases. On the other hand, you also have this absorption process. In the mouse brain, at the shorter wavelength, blood absorption dominates. At the longer wavelength, water absorption dominates because the brain is about 80% water. Okay? So if you put those two processes together, you have the exponential decay due to absorption. That's this curve. You have the exponential decay due to the scattering. Okay? That's this function. Put these together, these two things multiply together. And that means you can define a new attenuation length of your excitation process. That's the effective attenuation length, absorption length, and scattering length. Right? You put them together, that's this function which looks like this. It's a very crude model, but on the other hand, you can see it works pretty well. This is these two curves coming together, showing you like this. The smaller the number, the better. The smaller number means the longer the attenuation length. So these three dots are experimental data. Okay, so even with this very crude model for the mouse brain, and these three experimental data, the 800 nanometer, 1300 nanometer, and 1700 nanometer, they work pretty well for this crude model. But that means what? That means if you want to go deep, it's no-brainer to go to this window, which is roughly at the 1300 nanometer, or go to 1700 nanometer, because the attenuation is much less in the tissue. There's this little bump over here, and Jixing also mentioned, is the water absorption at about 1500 nanometer. So we started doing 1300 nanometer work first. We use the OPO. We look at the mouse brain. So we open a cranial window, replace the bone with a piece of cover glass so you can look through the top of the brain. We inject the dye over here as a Lexus 680. It's a long wavelength excitable red dye. And the blood will circulate so that the vasculature in the brain are all stained. So the blood becomes fluorescent. It's red fluorescence. So we use 2-photon excitation, 1300 nanometer. We just image down from the surface of the brain all the way, see how far can we go. And it works pretty well. As you can see, this movie going as a function of depth. So this is the depth. These are the blood vessels. You can see horizontal vessels is a line. Vertical vessels like a dot because you look at the horizontal cross section. And you can go pretty deep in this case. So we choose an area in this case where the gray matter, this is all in the mouse neocortex, the gray matter. But as you go in deeper and deeper, as you can see, the contrast gets reduced. And that's the signal to background ratio we talked about. Okay? So now we can quantify this movie by doing a three-dimensional reconstruction. Okay? These are the blood vessels in the mouse brain. You can see the capillaries. The capillary is about 3 micron diameter. And you can see different sort of lateral frames at different depth, right? So if you want to look at the fluorescence as a function of depth, okay? So remember the exponential decay due to absorption, due to scattering. If it's exponential function, if you do a semi-log plot, so you plot the log of the fluorescence as a function of depth, you expect a straight line decay, and the slope of which will give you the decay length, right? The slope. So you can see we determined at this wavelength, the decay length is about 285 microns, which actually is about factor of 2 better than 800 nanometer, which is very quantitative here. But on the other hand, you do see as you go deep and deep, eventually you can see this curve plateaus. That's exactly what the background limit is. If you do more quantitative, you can look at a signal to background ratio. Since only the blood vessels are labeled, that means the fluorescence should come from your blood vessel. Everywhere else supposed to be dark. But if you have background, that means everywhere else also gets somewhat brighter. So the signal is what we got from these blood vessels. That's a signal. The background, we just take pixels outside the blood vessel, which is supposed to be dark. If you quantitatively plot this as a function of depth, as you can see, for 2-photon, it does go down to almost 1 or 0. So it does go down as a function of depth. And we do reach this so-called SBR, signal to background ratio limit. And you can also do calculation to show that it works pretty well, roughly the same depth, about 5.5 scattering length into the brain. That's where you reach the fundamental limit. There's no hope to go any deeper, okay, as long as we're talking about 2-photon excitation. All right? So the second strategy we use is how to break down this fundamental SBR ratio limit, is to go to a high order process. Just like as I showed you before, why 2-photon is better than 1-photon. This is the same logic why 3-photon is better than 2-photon. So if you look at the axial profile of 2-photon excitation and 3-photon excitation. For 2-photon excitation, you can see you basically get your fluorescence from the focal plane, and the fluorescence decays as a function of 1 over z squared, and this z is the distance away from your focal plane. So it goes down as 1 over z squared. That's a classic second order 2-photon excitation. If you do 3-photon excitation, because now for 3-photon process, the signal goes as intensity cubic. So it's one order up. And that means it decays away from the focal plane much steeper. So in this case, it decays as 1 over z to the 4th power. Now, this actually, even as a graduate student, I already saw this. I actually wrote in my book chapter, my thesis said, this is great, much more axially confined. But this point, without scattering, as you can see, is kind of a no point. It's a moot point, right? Because you can see this is 10 to the minus 6. This is 10 to the minus 10. For imaging, you never get that kind of a SBR ratio. It's essentially comparing 0 to 0. There's no advantage here. But once you put scattering there, because remember we talked about, you have this exponential decay due to tissue scattering. And now if you put the exponential decay function on top of this focusing profile, you can see now this suppression of out of focus background is really meaningful, right? Because now this becomes appreciable far, far away from your focal point, all right? So if you quantify it, you can see you can actually suppress this by orders of magnitude. That means by going to the 3-photon process, we are no longer limited by the signal to background ratio, at least at the depth of, say, 2 to 3 millimeter depth in the brain. Okay, so we don't have this fundamental issue anymore. And in fact, For 3-photon imaging, as you can see, we are completely limited not by the SPI anymore. It's just by how much signal you can actually generate at the depth you have. So you can easily go to 2 millimeter or more without being constrained by this. Okay? So that's a two strategy. And it turns out, if you want to go to long wavelength, the 3-photon, actually the combination is perfect. Because the longer the wavelength you go, you can only access redder and redder dyes, right? Because that's what long wavelength does. And it turns out most dyes works in the visible. That just 90% of biologically relevant dyes works in the visible. The red dyes are there, but they're far and few in between. So if you want to combine the long wavelength window with 3-photon, that's quite perfect, because with 3-photon excitation, you can actually access a lot more molecules, because you can see for the same energy band gap, you have 3-photon combine their energy. That means you can reach much higher energy levels. Okay? So if you look at this, there's certainly a number of red and orange dyes can be 3-photon excitable at about 1700 nanometer. That means 1 photon absorption at the wavelength greater than 550 nanometer. Right? There are quite a few organic dyes available. There's also even better is that you can also use orange or red fluorescent proteins. Right? You can see all these molecules available. If you want to do 2 photon at 1700, you essentially have nothing to look at. Okay, just wavelength is too long. So the key question to make this 3-photon practical is this. This is what I did as a graduate student. You can see 1996. We already showed 3-photon is possible. There's no problem doing 3-photon microscopy. But in this case, you can see this is cell imaging without any tissue, just a single cell under the microscope, looking at DAPI-stained DNAs. Right? Even the sample surface, a transparent sample, with a titanium sapphire laser, we already use something like, it says, like 50 milliwatts average power. With that kind of average power, there's no hope of going deep, because your signal goes down exponentially. This is like at a zero depth, right? It's just a single cell. The key thing to realize is that it's a nonlinear process, as we talked about in the very beginning. The nonlinear process, for example, for 2-photon process, the signal goes inversely proportional to the duty cycle, repetition rate, and pulse width. If you do 3-photon excitation, the signal goes as the inverse duty cycle squared. Okay? And that means by reducing the duty cycle of your laser, just using a different laser source, you can actually make the 3-photon signal much stronger. See, this is what I did in graduate school. You can see we worked around here with the titanium sapphire laser. Of course, you can see the 3-photon signal is down by more than 2 orders of magnitude. It's not practical. It's doable, but why do you want to do it? But if you use a duty cycle over about here, 2 orders of magnitude down, you can see the 3-photon signal and 2-photon signal can be quite comparable at average power of a few milliwatts. So the key thing is to have the right laser to do this technology. So what you need is roughly about a megahertz laser, which still allows you to do reasonably fast imaging, but with high pulse energy for the 3-photon microscopy. So since I do fiber optics, so we actually look around to develop this new laser source at a 1700 nanometer. Of course, as I always say, whatever wavelength you want, you cannot find a laser. That's usually what happens. Right? Laser is there, but not the wavelength you want. So we set out to using fiber optics to develop a new source at a 1700 nanometer. And the technology we use is called, connected to Jixing's talk, this Raman effect. So as we all know the Raman effect, a ground state molecule excites to a virtual state, and the molecule can actually decay down from the virtual state and a Raman photon. And because of that, you can see this photon, the Raman photon, typically is Stokes shifted. So it's rest shifted from your excitation photon. Now we can use this process in optical fiber. In that regard is that you can see using this Raman process, you can see a short pulse will have a broad spectral bandwidth. That means if you launch such a pulse into optical fiber just SiO2. SiO2 has its Raman gain, Raman excitation. Using the Raman excitation, you can see the blue portion of this pulse can actually pump the red portion of this pulse through this Raman process. Okay? The net effect is that this pulse will transfer its energy from the blue to the red. And that means the spectra of the pulse will shift to the red. That's what it does. And we use a fancy method. Yeah.
Is it possible that the red line further that the fiber and further red?
Exactly, continuously redshifted, absolutely. It will continuously shift. That's exactly the. No, it won't, because when the blue pumps the red, the blue actually will lose its power. That makes sense? The blue actually will lose its power.
The red line will further affect
Yeah, the red light will, okay, so this, essentially you can see this will lose to here, right? So the pulse will shift to the little bit like this. And that means, and then this will pump this again, will continue to shift. But we use this called a soliton effect to maintain the spectral profile. So where the soliton? Yeah, the soliton effects to maintain the temporal and spectral profile. So that a short pulse coming in, we have a short pulse coming out. Okay, so the soliton effect is very important. If you don't have the soliton effect, this won't happen because the pulse will just broaden as it propagates. And this technology really works. This is what I did at the Lucent Technologies, many years ago. We, for example, shining in a 1300 nanometer light into this so-called grapefruit fiber, looks like a grapefruit, and we shifted all the way to about 1700 nanometer. So the idea really works. But of course, at the time, we even patented this, by the way, because they give me $1,000 every time I patent something. So, but the pulse energy is really too low. Okay, the pulse energy for this kind of technology at the time is only 0.1 nanojoule. Normally, your titanium sapphire laser is about 10 nanojoule. And what I need is 100 nanojoule. You can see it's about 1,000 times less than what I need. But the good thing is that in the last decade or so, you can see all these different fibers coming online. You can just go buy it, right? And using different fiber platforms, you can do the same soliton shift idea, Raman shift idea, but with a variety of different pulse energies. Right? So this, for example, called large mode area fiber, you get pulse energy about 10 nanojoule, like your TiSAF. This is photonic crystal rod, which is what we've been using, is about 100 nanojoule pulses, which will be useful for 3 photon. And if you want to go to air core bandgap fiber, you can go to pulse energy close to a microjoule. So depending on the platform you select, you can get the pulse energy you want and still maintain this soliton shift idea. And this is an example of soliton shift as you asked. You can see, depending on the power I launch into this fiber, I can shift the soliton further and further in the red. It's a continuous shift. In that regard, you can see there's also secondary solitons coming up due to the residual power in your input. Okay? So you can see the soliton can shift more and more to the red by putting in more and more input power to the fiber. And by doing so, you can see we can form lasers by shifting something from, say, 50 nanometer, which is a very nice wavelength, because of telecom, because you have lots of lasers available over there. So the fiber we use is called photonic crystal rod. Essentially, if you look at outside, it's just like a glass rod, about 1 millimeter diameter, nothing special. Of course, it costs $10,000 to begin with, and also the detailed microstructures built into this. You can't really see from the outside. So this is a numerical simulation just to show that we do understand this process extremely well. By the way, it's a telecom simulation tool, so it works extremely well, right? So you can see this is a wavelength domain. This is a time domain, right? So we launch a 400 femtosecond pulse. The pulse first gets compressed in the glass rod. This is plotting as a function of propagation distance into the rod. You can see the soliton coming out over here and shift to the long wavelength. This is the soliton in the time domain over here, right? So you can see we can shift the soliton to about 1700 nanometer in a rod about a foot long, just a glass rod this long. And in addition, I launch 400 femtosecond. I compress the pulse to 66 femtosecond because of the anomalous dispersion soliton compression process. And then we get pulse energy about 67 nanojoule. So the peak power is really a megawatt. So this actually is by far the highest pulse energy I guess human being has obtained from a waveguide structure. Okay, a megawatt peak power. And this is some freeze frames. So you can see the input spectrum, 1550. This is the laser. And shifting to about 1700 over here after this photonic crystal rod. In the time domain, you can see coming in at 400 femtosecond, coming out at about 66 femtosecond. And this is the microscope we use, very crude. You can see scan lens, tube lens, and objective lens. Now to use something at this wavelength, you actually have to engineer all those different optics, because regular microscope, you put in this wavelength, you have very little coming out of it, right? Because it's, the transmission typically is really bad for this. But that's not a hard problem to solve. So this is showing you in vivo imaging of the mouse neuron labeled by red fluorescent protein. It's called a rainbow mouse because the neurons in this mouse are expressing different type of fluorescent protein. So they show up like a rainbow color. Again, as a function of depth, as you can see. So you can see the blood vessel here is the blue. The third harmonic actually showing you the blue blood vessel. And the red thing are the neurons. Okay, this is the depth. This scale bar is 50 microns. As we go down, we increase the laser power to a maximum power of 20 milliwatts at the brain surface.
What are the dark areas?
The dark areas are neurons not labeled. Yeah, because not every neuron expresses that protein. And you can see the myelin sheath, the white matter, very clearly, the striated portion, and it actually changes the orientation for the myelin. That's actually something interesting. So we can actually, for the first time, see something in the hippocampus. Okay. So I just show you the three-dimensional reconstruction over here, and you have, for example, this. Oh, okay, that's good. My time haven't changed. I thought, oh my God, I'm way over time. But this is still Eastern time, no problem. Yeah. So these white dots are the neuron bodies, the soma, and these thin things are the neuronal processes like the axons, the dendrites. And this is the histology. So you can see the SP layer of the hippocampus. And this is the rainbow mouse. We image roughly this region, and this is the anatomy. And this is the lateral features, lateral frames, that different layers of neurons. You can see the different layers of neurons all the way down to hippocampus, passing through the white matter. And the white matter, as Jixing also mentioned, shows very nice third harmonic generation of the myelins. So you can see the nice myelin structures in the white matter. Okay? So in the beginning, we actually worry about how do we know we hit the white matter. But it turns out this is a great feature. Every time you hit the white matter, you cannot miss it. The third harmonic is very clear to be visible. And just to show that the resolution is not bad, even at the great depth, we have sub-micron resolution laterally, and we have about 5 microns Z resolution, the sectioning resolution, even at about a millimeter into the brain. So still very decent subcellular resolution of the neuron. And just skip that. So in the last year or so, after this demonstration, we know we can look at the neuron very deep into the brain. But what we really want is look at neuronal function, right? The reason to imaging mouse brain in vivo, an intact brain, is to look at how the brain functions, how the neuron fires. And the way we do neuronal activity recording is to using calcium imaging. Essentially, every time a neuron fires, the channel will open, calcium will rush in, so the intracellular calcium concentration is going to increase. And you have these proteins engineered to respond to the concentration of the calcium. And these are called the calcium indicators. So you can use organic dyes, like calcium green, calcium orange, fura. You can use genetic engineered fluorescent proteins, such as GCAMs, green fluorescent based. And it's been proven a long time ago. I just showed you Raphael yesterday's paper more than 10 years ago, showing that every neuronal action potential spike corresponds to the calcium concentration jump. Okay? So this is the potential measurement of a neuron. This is the fluorescence measurement called a delta F over F. Delta F is how much fluorescence change over the baseline fluorescence, F. You can see every spike you have, it shows a calcium transient. So by imaging the calcium concentration, you can actually faithfully record each action potential. Well, it's the same. It correlates to the action potential. Now the problem for that is these calcium indicators are only robust for in vivo imaging in the grain. There's actually no red, orange calcium indicators currently available for in vivo imaging. You have to go back to the green dyes, calcium green, for example, GCAMs. There's no red things. Now the red fluorescent protein, there's people are working on this very feverishly, but I still think it's about a generation or two away, at least to be compatible, to be comparable with those very nice calcium indicators. But the problem with the dye development is really, I'm kind of pessimistic always. It could be 6 months, it could be a year, it could be a decade or two. We don't know. Eventually they might come, but when they come, you get it. If they don't come, you just have to wait. So what do we have to do? So what you have to do is, how do we combine these long wavelength spectral windows at 1300 or 1700 with the green probes? But the green probes are great, People engineer that for about a decade. They really are awesome. Okay? So how do we combine this? What's your idea? It's actually pretty trivial, right? How do we combine 1300 and 1700 with the green dyes? There's basically only two ways you can do it. One is 3-photon excitation at the 1300 nanometer. If you do that, you can see you can access 1-photon fluorophores with excitation around 430. Now that's blue, which means green emission. That's doable. You can also try 4-photon. Why not? Right, at the 1700. That will also give you roughly the blue excitation. So we can try both. So I'll show you some new results. We're trying both of those things. Now, if you look at 1300 nanometer imaging, what's the advantage for that? The biggest advantage is really the top two. You access the green fluorophores. Again, 90% of the biologically relevant fluorophores probably in the green, in the visible. And actually, because of the shorter wavelength, for 1300, you're actually allowed to use higher average power. That's actually very appreciable. If you want to image deep, average power also matters. Okay, so you have two advantages. The disadvantage for using 1300 compared with 1700, oops, I touched something accidentally, is that its decay length is slightly shorter, so you exponentially decay a little bit faster, which is not as good. And this excitation source is very cumbersome. But on the other hand, like Steve and I, we developed those sources. That could be a great opportunity. You can pitch for $1 million to develop those things, right? But bottom line is that, from my calculation, within those depths, those two techniques are quite comparable within a factor of 3 of each other in signal generation. So if you only concentrate about a millimeter or so in the brain, both wavelengths will work very well.
Yeah.
So is there like a limit that you know for the peak power that beyond?
Oh, that's the limit I put down. I know at 140 milliwatts at 1300, it's safe. No acute damage.
If it was higher, like 400, let's say 400 should still stay.
Yeah, that's the same. Yeah, the average power is the same. If you're interested in the damage, I have a lot more details. If I can, I didn't include it today. So just show you one example we started doing is we bought this genetically engineered mouse with a GFP labeled neuron, very sparsely labeled. That you can just buy from Jackson Lab. So you can see at 1300 nanometer 3-photon, we can actually go also to the hippocampus about 1100 microns in. You can see nice features of the Y matter, third harmonic. You can see neurons at different layers. It's a sparsely labeled, so it doesn't look as pretty an image. But just to show that the depth capability is still there by going to 1300 nanometer. Of course, the goal is to looking into brain activity using GCAMs. Because if you can do GFP, you can probably do GCAM. Okay? Okay, this is something you sweep under the rug. That's a laser you have right now. Not very practical, costs about $400,000. If you happen to have it, you might as well use it, right? So we use this laser. We are developing something better than this. But that's what you have. That's the best you have. So anyway, skip that. So now we can look at neuronal activity of GCAMP labeled neurons in the cortex. So this is the third harmonic image showing you the Y matters. In this mouse, the Y matter is a little bit shallow at about 700 microns starting. So you can see the neurons at different layers using the GCAMP label. And this is the neuronal activity we can record from the soma. You can see the fluorescence as a function of time. Okay, and this is the activity recorded from the dendrite. You can see, you also see those transient of the calcium concentration. And this whole trace is about 5 minutes. Now we actually come back to image the same neuron 3 times over the course of about 2 weeks, and we detect no change for the neuronal firing. Just want to make sure there's no damage. At least from that standpoint, there's no damage. I'll show you a movie for this. You can see this is a neuron. You can see the flash of the calcium signal. You can see it follows this trace roughly. It gets brighter, it's the peak. Right? You can see the neuronal activity for the body, for the dendrites. Right? You can see it lights up. Now it's probably going to here, so it's kind of quiet. A brief flash, probably this one here. Now it's quiet again. They'll flash again in the end.
So there's a low decay signal. Is that really the calcium decay?
Yeah, the calcium dyes, it really is a problem. The dyes are slow. So this is GCAMP-S, the GCAMP slow. It's a characteristic time, yeah, that's what it means, is half a second. But you have GCAMP-F, GCAMP fast, can go maybe about the 200 milliseconds. But that's a problem for calcium imaging. You don't have the single spike capability because it's very slow. it kind of integrates. So it's hard to, membrane potential dye can do better. But membrane potential dye, you can see these spikes are huge. You're talking about 100% intensity change. So you can watch this in real time on a microscope. You can just see it by eye as you go. This is about 700 microns in, layer 6 neurons. So the deeper you go in the cortex. With membrane potential dye, you only have a 1% change. It's very hard to see. Okay. So that's the movie. And I promised we can also try 4 photon, right? 4 photon 1700 can also get to the grain. And 4 photon also works. You can see 3 photon Texas red, 4 photon fluorescing. Fluorescing is a green dye, also works. So 4 photon does work. But the only problem for 4 photon is that every time you go to high order, as we talked about, you want to get enough signal coming back. The drawback always is that you have to go to reduced duty cycle or reduced repetition rate because the pulse width is already down to 50 nanometer or 50 femtosecond. It's hard to reduce it much further. To get enough signal at the high order process, you got to use lower and lower repetition rate. So that's the sacrifice you make. Going to 3 photon, 4 photon, the sacrifice is you image somewhat slower than 2 photon microscopy. That's a compromise you make. But on the other hand, it does allow you to go deep, right? So 4 photon does enable you to go imaging green fluorophores like GFP, for example, and you can do that. I'll just show you one example. We haven't got the activity going. Just showing some examples of the GFP fluorescence, 4 photon fluorescence, and a third harmonic generation, still 500 microns in the mouse brain. Okay? And even more exciting, I think, this very recent results we did in the mouse heart. Heart is actually very tough to image deep, but this time we can go about 2 to 3 times deeper as a regular 2 photon microscopy. And even better is we can do 3 photon red dyes and 4 photon green fluorescent protein. And by doing so, you can see you can do multicolor imaging with 1 wavelength, right? So these are the heart muscles. You can see 3 photon fluorescence of Texas red, whoops, and the 4 photon fluorescence of GFP. You can merge them together. And of course, if you can look at GFP, I'm very confident we can also look at GCAMs. So potentially you can also try that. The last thing I want to mention is we want to, for example, do awakened behaving animal, right? You know, all the animal we look so far is anesthetized, sleeping. And if we're looking for brain activity, sleeping thing is not very interesting. Might be dreaming, but still not very interesting. So we can do 2 things. One is those miniature microscopes I talked about in the very beginning. We do. So we could potentially mount this on a mouse head. These things only weighs about a couple of grams. So it's light enough to be on a 30-gram mouse, for example. But a little bit easier is this. What we did is we copied the setup from David Tank's lab from Princeton. We can make a spherical treadmill, and the mouse can actually run on a treadmill in a head-fixed condition. So we can look at the brain, for example, over here, while the mouse running around on this ball. So that's actually possible. So we have this already going. So how much time I have? 5 minutes? Okay, so I just briefly mentioned, okay, how do we push the limits? Like we show we can go deep, we can show we can do imaging activity and others. One thing obviously can go deeper is to develop an optimized excitation source. In some sense, a laser source is really like an engine of imaging. You really have to optimize the situation to drive your imaging system. So there's some detailed analysis going in, what kind of laser one want for multiphoton imaging. But in this case, I just skipped some of the detailed logic, just give you the final take-home message. So the bottom line is that I think for the optimal repetition rate for deep tissue microscope is about 5 megahertz for 2 photon imaging and 1 megahertz for 3 photon imaging. If you want to go deep that's all in the context of deep imaging. If you want to imaging surface, that's totally different. If you want to go, say, 3-4 scattering depth into the tissue, and that's the kind of a system you want at much lower repetition rate, the 80 megahertz everybody uses. In some sense, the 80 megahertz is artificial, right? Just because the box is 2 meters long, that's what it is, right? There's no other reason for 80 megahertz. But if you want to develop something optimized, because you want to push the limit, this is much better. And just to show we can get a much better source than what I showed you before, is that we actually even got some bigger photonic crystal rod. We can now get about, instead of 1 megawatt soliton, we can get almost 3.5, 4 megawatt solitons coming out of this photonic crystal rod. And very interestingly, as you can see, this peak power actually can go beyond the critical power of self-focusing. That's one thing I worry, because every time you damage those things, it's $10,000, right? But at least we tried it, nothing happened, because it actually makes sense logically. In a waveguide device, the confinement of the waveguide overrides the focusing. So it's not surprising to see that, but still a little bit worrisome because it's a $10,000 experiment, right? So I think it's achievable to develop those sources. And I also talked about at 1300, it's a great opportunity to develop a new source to replace this $400,000 OPA amplifier we have. And signal collection, I think, is another opportunity. I think all these objectives are designed for regular microscopes. It's really not optimized yet for deep tissue penetration. So you want to collect signal better. I think there's room to improve on this. And we developed some of those things with Olympus together. It costs a lot of money, but it works better. That's roughly the cost of these lenses, right? But at least I think it's possible to collect efficiently even at a depth about 2 millimeter in. It's definitely possible. It may require a couple more of those, right? But it's at least possible to do. So if you want to get a factor of 10 improvement, it's probably possible still. So there's something more to do there. And we also now jump into this thing called wavefront sensing. We haven't any data to show, but we're very excited. So this is the setup in the lab right now, exactly the ZMAX drawing of what we have. So we try to compensate for waveforms, optical waveform in the brain. And there's a lot of debate on what are we compensating. Are we compensating for aberration? Are we compensating for scattering? But it doesn't really matter, right? Because I know for sure by simulation, for 3-photon excitation, this will work much better than 2-photon, just because of high-order nonlinear process. And it will help you in two regards. One is you can achieve much better focus. You can perhaps resolve the dendritic spines if you can do adaptive optics down in the brain. So half a micron or less resolution possible. And second, maybe even better, is it can probably enhance your signal. So we think we can get at least factor of 10 signal enhancement, maybe upwards of 100. Okay, so that can really help in the deep tissue imaging. So that's what we're doing. So the ultimate goal, we hope, is to do this type of stuff. We want to image about 1 millimeter cube of brain tissue exhaustively. Means we want to sample something at about 100 megasamples per second. If you can do that, you can more or less sample this 1 millimeter cube of brain tissue in exhaustive fashion of every spike, if you have the sampling rate of this. So here's what I think we can do by just putting things we have together. We can do roughly this. At a millimeter depth, we can roughly do about a megasample per second. That's what we can roughly do. And I think we can already perform many interesting measurements, even at this kind of a level. At a millimeter depth, at a hippocampal level, at about a megasample per second. There's many biologies you can do already in this kind of system. But to be exhaustive, you're still about 100 times too slow. Of course, if you want to go down to 2 millimeter, you can see. You want to go deeper, your signal is less. You can maybe only do a kilosample per second. Now that's way too slow for most biologists. Okay, I think this is roughly the minimum you want to achieve to look for interesting biology. And this is way too slow. So we hope is we can throw this wavefront correction into the mix to make up this about a factor of 100 for us. And hopefully it'll come up soon, maybe a couple of months. We've been working on this for a while already. So just to summarize my talk, I give you some motivation to bridge the microscopic to the macroscopic using optical imaging. And the long wavelength approach and the high order 3-photon approach is superior if you want to go deep in the brain. And I talk about these 2 spectral windows and 3-photon microscopy. And we can image the brain activity by using the green 404 and using 1300 nanometer 3-photon microscopy. And we can do a combination of 3-4 photon process. And I'll give you some future directions, such as new sources, new objectives, and maybe adaptive optics, compensation method, to push this imaging further. So thank you very much.
Question, Tim?
So I was wondering two things. So your goal is to image in vivo brain activity.
Yes.
But it wasn't the case. For example, there are optical clearing techniques out there to reduce the scattering.
Absolutely.
I'm wondering how much that might improve further.
If you do optical clearing, if you want to do ex vivo, the optical clearing really works well. So in that case, you don't need any of those, because it's like imaging to water, right? If you have like a clarity, scale of your objective, you can go 4 millimeter, 8 millimeter into the brain. But the only issue is that if in the brain context in particular, it's really important to look at intact brain in vivo. Because if you take a neuron out or you slice the brain off, the activity probably not as interesting, let's put it that way, right? You cannot do behavior studies and things to correlate. So I think if we can do clarity, then none of these matters.
And the second question was basically, I'm wondering how, so you mentioned at some point, it all comes down to the point that how much photons you can excite the fluorophore with. So I guess it also matters how good the fluorophore is.
Yeah.
And so what do you think that quantum dots, let's say?
Yeah, quantum dots, for example, in terms of cross-section, quantum dots are at least 100 times brighter than fluorophore. But on the other hand, it's a bit of cheating, right? If you think about it, a fluorophore has a molecular weight of 500. Has a size maybe a few angstroms. A quantum dot has a much bigger cross-section, yes, but it's a much bigger entity. It's 5 nanometers. So in that regard, it's a larger cross-section, but it's sort of an artifact of the density of states. You just have a lot more molecules. So all the states sort of jam together to give you a bigger cross-section. That makes sense? It's a much bigger entity, that's what I'm saying. So in terms of one molecule to one quantum dot, yes, quantum dot is much brighter. But on the other hand, if you're imaging in vivo, the dyes can go much higher concentration than the quantum dots. That makes sense? You can put more dyes into the blood than the quantum dots. So in that case, kind of comes back. The depth. Yeah, basically in this case it's all simple. We basically start the brains, actually we start the deepest. We put the objective in, right? We just keep the mouse the same, we lift our objective length up. Because the multi-photons are three-dimensionally confined, so you look at your motor translation, you know exactly where that is. How do you express those proteins? Okay, so for some of the mouse, like a Brainbow and this GFP mouse, we just bought it from Jackson Lab. They engineered it for us, right? So that means you actually transfect the babies or something. I don't know exactly how they do it. But for the activity, what we have done here is we actually inject virus into the mouse brain. So these are GCAMP called AAV virus. You inject directly to the site you want to image. And it will infect the neuron and express the protein. It's not the best, but lately you can see these mouse will become commercially available very quickly, which would be great for us. The virus injection, by the way, I didn't tell you, it took us 6 months to conquer. Simple as that. Everybody says, just a virus injection. But when you do it, it takes 6 months. Without label, if you do autofluorescence, we actually never tried autofluorescence. Okay, autofluorescence for these things is very bad. For the multi-photon, it could be good and bad. The good thing about this is that we don't see much autofluorescence, which could be great if you want to look for exogenous dyes, so you don't have background. But the bad thing is that you probably cannot do much autofluorescence. So we tried autofluorescence at the 1060. That works 3 photon. And then DH and all those. At 1700, that's a good thing, could be, because you don't have background.
So in the mouse brain, how far apart are the cell bodies of two interacting neurons?
How far apart they are, could be. I don't know. In the z-axis, I thought the neurons actually, you guys probably know better than I do. The neurons actually connect vertically, right? You have different layers of neurons. They break both ways? Yeah. So you have these different layers of neurons. At the Y-matter, actually, supposedly, these myelinated axons can transport the signal in macroscopic distance. You're talking about millimeters. So it could be extended. Yeah. But the computational columns, supposedly, if you can do 1 millimeter cubic, you can probably isolate those computational columns, neuronal columns.
I guess I have one other question too about the wavefront shape. That sometimes can be quite slow too. Are you trying to do shape dynamically as you scan?
Yeah, this actually is very interesting. If you look at this, what are we compensating? I mean, I think if you correct for aberration, you probably don't have to worry about too much dynamics. That's why it could be practical. If we go into this regime, it's not going to be practical in a sense that blood flows, and when in vivo, things move. I mean, you can see the movie I showed you, the shift, because the mouse sometimes just moves, right? So this probably be very tough. But this, because of the blood vessel, for example, never changes, cell body probably where they are, and you don't mind, say, half a micron shift. So this will probably give you more play. So essentially, it's hard to, the transition is really it's really the order. So I don't think we add yeah. But in some sense, it's not going to matter.
All right, thanks. Keep going. 5 minutes.